In 2002, Professor Thomas Meade moved to Northwestern University to direct the Center for Advanced Molecular Imaging, part of the Chemistry of Life Processes Institute. At this multidisciplinary institute, Professor Meade and his colleagues explore molecular imaging, transcription factor inhibitors, and the development of electronic biosensors for the detection of DNA and proteins. Tonight, Professor Meade will discuss his work in molecular imaging and the role metals play in this emerging field.
Greetings. My name is Professor Meade, and I'm broadcasting from my laboratories in the Chemistry of Life Processes Institute here at Northwestern University. I'm glad you could join me tonight to learn more about what we do with molecular imaging and how the metals play such an important role in our research. Imagine that I put before you a rack of 50 ml conical tubes filled with opaque purple jello. And I further tell you that in each tube there might be grapes or grapes with seeds. Your task is to figure out which tubes have the grapes with seeds. Since it's opaque, you can't see through them. You can't tell which ones are which. So I'm going to give you 3 tools. The first tool is a scalpel, where you'll take each individual tube and dissect it. And when you find a tube with a grape, you'll further dissect that grape to see if it has seeds. The second technique will be a vat of acid, where you're going to dissolve the tubes. And while they're dissolving, you'll carefully watch for those tubes that have the grapes with seeds. And finally, you're going to take your rack of tubes and you're going to put them in a magnetic resonance scanner. And in that scanner, we're going to take a 3D image, which will show all the tubes in one shot. Now, each of those tubes there with that 3D image haven't been touched. So you have an intact sample when you're done. Imagine that those grapes with seeds are the cancer. Imagine the grapes just being organs or other parts of your body. And finally, the purple jello by itself is just normal tissue. Which technique of these 3 would you like to use to find cancer in you? I think the choice is pretty obvious. You're going to rather have a magnetic resonance scan where I didn't have to cut you up or dissolve you to find the cancer. So, what can molecular imaging do? Molecular imaging provides researchers and clinicians the ability to visualize and investigate complex biochemical phenomena in their natural environment. These are intact, living organisms that we can visualize images in real time with no need to slice or dice the subject. It exploits the use of exogenous, meaning injected, imaging agents. And I want you to think dyes. And these dyes can be targeted or bioactivated to enhance the kinds of information acquired. There are many modalities or techniques that we use to scan. I want you to think of your first dental exam that looked for cavities, or perhaps when you broke a bone. That modality, of course, was X-rays. But there are many more modalities. One of the prime examples is optical techniques, microscopes that are used for high-resolution cellular imaging. Ultrasound, used in pregnancies to keep an eye on a fetus while it's developing, and many other applications. Magnetic resonance imaging. This is excellent for soft tissue, like your brain or other major organs. And finally, nuclear imaging. For example, positron emission tomography, which is extremely sensitive for detection of tumors. Each modality has distinct advantages and disadvantages, or limitations. Specifically, in regards to spatial resolution, or sensitivity. So, which modality is the one that should be used? Well, that depends. At the outset of any imaging experiment, a couple of questions must be asked. What is the spatial resolution and level of sensitivity required to answer the scientific or clinical question? For example, how small is the target? Is it a single cell or is it a whole organ? How much of the target is present, like the concentration of a receptor in the body? The answers to these questions can help you decide which modality to use. Optical imaging has excellent spatial resolution, submicron, meaning we can see inside single cells. But it has limited depth penetration for looking inside whole animals. While magnetic resonance imaging has unlimited depth penetration, it has limited spatial resolution. This means you can only see things 100 microns, as opposed to submicron with optical. You can't see single cells, but you can obtain a 3D image of an entire animal or a patient. So now that you've chosen your modality, another component to consider is using an imaging agent. And this is where the chemists come to the table. These exogenous contrast agents, or dyes, are injected into the subject and help delineate anatomical features, such as disruption of the blood-brain barrier, lesions such as tumors, infarcted tissues, or vascular injury. So how do imaging agents work? Well, some of you may know what a septic tank is, and some of you may have also experienced one that is leaking. You carry a detector with you at all times that lets you know when that tank is leaking. And I think you know what it is. Your nose tells you there's a leak, but it doesn't tell you where that leak is. So you call Roto-Rooter folks, and they come out with 3 items: a lawn chair, a newspaper, and a can of fluorescent dye. They flush the dye down the toilet, sit in the yard, and wait for the dye to percolate to the surface. And that's where they know to dig to fix the leak. That's what contrast agents do. You have blunt trauma to the head. They inject a contrast agent, put you in the scanner, and wherever the dye is not supposed to be is where the problem is. So what are the elements from the periodic table that make up contrast agents? For PET imaging, you have carbon-11, nitrogen-13, fluorine-18, oxygen-15, copper-64, and gallium-67. And all of these elements have a radioactive signature, which the PET scanner can detect. On the other hand, MR imaging uses gadolinium, manganese, and iron nanoparticles, where they relax certain tissues. For example, in an MR image, the blood is very bright, whereas other tissues and parts of the brain or other organs can be darker. But what's happening now? In my lab, and many others, we are developing what we call smart contrast agents. Unlike traditional agents that only provide anatomical information, smart agents detect biochemical and physiological processes. Where a traditional agent can't detect enzyme activity or pH, these agents can. They accomplish this by not only reporting on their location, but can be activated by a specific biochemical event, like an on-off switch, tuned to a specific physiology that we are interested in. Let's take an example. Correlating gene expression in whole animals with developmental biology is crucial to understanding how life works. Currently, you image the gene expression by sacrificing many animals at different stages of development, then fix and stain to see where the gene expression was. It's sort of like finding the grapes with seeds. We're back to using scalpels and acid. But now, with smart agents, we've turned this process into a 3D movie, where we simply visualize the gene expression in the living body, in real time. No slicing or dicing. In one experiment, we used beta-galactosidase as a reporter gene for a developmental process in amphibians and mice. We injected the agent into the embryo, and as the animal grew, the agent was turned on at specific developmental stages following the gene of interest. When that gene is activated, it turns on the agent, and we see it in the image. So what can molecular imaging mean to you and your future? This is a very young field, impacting basic science and clinical translation. So whether you're a chemist, a biologist, an engineer, or a pre-med student, molecular imaging has a future in your future. We're currently set to explore and discover biological mechanisms that we couldn't imagine addressing not long ago. What if I were to tell you that you can take a molecular imaging agent, a diagnostic, and attach it to a drug to create what we now call theranostics, therapy and diagnostics in one agent. These agents are capable of diagnosing, detecting, and simultaneously treating diseases such as cancer and many others. Ultimately, molecular imaging is making clinical diagnoses more accurate and is becoming one of the pillars of personalized medicine. I want to thank you for joining us tonight, and I look forward to answering your questions.
Thank you, Professor Meade, and we look forward to your answers.